So one of the most errors that in questions I had was in semi-global alignment. So I answered some of your questions. You mostly forgot how to apply the terminal how gap penalty. Terminal gaps are not penalized. You didn't know how to. Some of you did not apply it on the dynamic programming partial scores table in the last row and the last column, and that was one of the main problems. Other than that, everybody performed nicely. Some of you had difficulties in timing. The time may not seem sufficient, but we did a little analysis on that, and the Markov model question actually, you had like 1 hour. If the true false, for example, 5 minutes, the short definitions, if each one of them is like 5 minutes, you should be able to do that. The short definitions, the true false in 5 minutes, the star alignment, if you spend 15 minutes on that, and also the semi-global alignment should not take more than 20 minutes of your time. If you add those up, I mean, the hidden Markov model question gets at least like 50 minutes out of this 110 minutes in total. So you should be able to do it in time. But in the final exam, I will try to give you more time if you have timing problems. Yeah, in general, the exam, everybody did very well in the exam. Thanks for your studies. And if you have any particular questions, I'll be happy to answer them before we start talking about phylogenetic trees. Or if you have any questions, you may also send me an email anytime you want. And the final exam is going to contain topics after the midterm, so it will start with phylogenetic trees. So the final exam is going to start from subtopics, including phylogenetic trees and onwards. After phylogenetic trees, we are going to talk about protein structures and then gene expression analysis. And for that, we're going to focus on the clustering problem, not differential gene expression, but the clustering. We are going to see clustering algorithms to find similar behaving genes in terms of their expression. And then the final topics we're going to cover in the last 6 weeks after the spring break is also going to include computational systems biology, where we are going to talk about biological networks and some analysis techniques on those biological networks and some graph theoretic measures like clustering coefficient or the centrality measures, some of them we are going to consider. Okay, so this is the plan for the rest of the semester. And I'm also going to share with you the solution for the second assignment tomorrow. All right, so you'll also have a chance to look at the solution. There's not going to be an assignment or any work for the spring break. So I want you to enjoy spring break and Ramazan Bayramı with no workload from this course at least. So we are going to have our third assignment probably on protein structures. So after we come to a certain point in protein structures, I will give you your third assignment. And it may be either a programming assignment of some kind, which will be working on protein data bank PDB files and to collect certain information from protein structures. Okay, so it may be a protein structure. It will be a protein structure related programming assignment. All right. If no questions, I will continue with the phylogenetic trees. And in the beginning of the semester, in my sample lecture, we actually talked about phylogenetic trees. If I remember correctly, I talked about this method, right? Additive trees from additive matrices. I described the problem. And also, let me change the mouse pointer size. I described the problem of constructing the phylogenetic tree, and we talked about this method where we solve a system of linear equations to find, to put, to add each node one by one to the tree and come up with the final tree. As you can see, this is a weighted tree where edges are weighted, and each of the species or genes we have are at the leaf nodes. Okay, so I'm going to quickly go over this method again, but let's go over the slides one by one and let me re-describe the problem, what we are trying to do. What is this phylogenetic analysis that we are doing? This is similar, related to also these multiple sequence alignment, the profiles we have learned. Remember, for example, cluster W was using phylogenetic tree as the guide tree. So when we have multiple things, multiple objects, these may be multiple genes, or these may be multiple species, they may be, so we maybe compare biological entities at the organism level, or at the gene level, or at other levels. So phylogenetic trees gives you a way to show you the relationship between those multiple entities in a hierarchical manner. So the methods that we are going to learn here can also be considered as clustering algorithms, as it will show us, for example, if you look at this, for example, a clustering of certain patients, samples that are taken from certain patients or local control together in a group. So this is like a hierarchical grouping of individual items, which tells you that, for example, local control 2 and 3 are more similar to each other than these one. They form, if you cut in another way, if you're given a hierarchical tree like this, if you cut this tree at a particular level, you will have a certain number of groups. For example, all these will be in separate groups, but these 1, 2, 3, 4, 5, 6, 7, 8. These 8 are in a single group. These 2 are in a single group, and these 2 are in a group, and the remaining single samples are in their separate groups. But a hierarchical clustering like this, a hierarchical grouping like this in phylogenetic trees also gives you a chance to cluster them with different granularities. If you, for example, cut it here, you see that the group of 8 now is divided into different groups. These 2 are more similar to each other. They're in a single group. These 2 are also they're single groups. Patient B and patient B is in a group together. So cutting this at different levels, if I cut it here, you'll see local control 9 can also be put into the same group as this one. So which means that you can play with the granularity and have a different granularity clustering, grouping of your individual samples if you have a tree, a phylogenetic tree, a dendrogram that of those individual objects you have. Okay, so we are going to see algorithms which are used to build such trees. And we will be focusing on trees where we build them using distance matrices. They could have been similarity matrices, which could be, if you have a similarity matrix, you can turn it into a distance matrix by just taking the inverse of those similarities or with just some function. So it's not so difficult to do that. But basically we are going to have some numerical measure of distance or similarity between individual items. By using that, you're going to build such matrices. There are also such dendrograms. There are also methods which work with non-numerical data or some vector data, which lists a list of features, whether those features exist in a species or not. So these are like character matrices. There are also like minimum parsimony. So this method, maximum parsimony method. So this character base phylogeny. We may have a character state matrix, for example. These individual objects, do they show these characteristics or not? There's also methods to build phylogenetic trees based on that. We're not going to focus on those methods in this course. Okay, in this course, we're going to, in this chapter, we're going to assume that we have an input matrix that is like this, which shows, by the way, these should be also capital A, capital B, capital C, capital E, and capital E. They are the same objects, which shows pairwise relationship between genes, organisms, represented by a single quantitative, some number, some numerical distance measure between them. Okay, so if we go this phylogeny, it's coined by 18 in the late 19, the second half of the 19th century by Haeckel. So when biologists try to understand the organisms on Earth better, they try to figure out relationships between them. So this classification, grouping them together in an organized manner is a need that is way before we had genetics, all right, before even these whole genomes appeared. 150 years ago, before whole genomes started appearing, we had the need for organizing these species. So basically, the line of the relationship between these species, they were put in these origins of life type of trees, okay? Tree of life, or which put plants, animals, or bacteria in different groups. So that's basically given also as a tree, which is a hierarchy. We have families, species, subfamilies in these organisms. So this is basically a need in molecular biology. And the goals of phylogenetic analysis can be, well, different things these days. We can understand the evolutionary history of human beings. We can understand, for example, the spread of a virus like COVID-19. We can actually look at different strains. We have these different mutations, different variants of these virus. We can just look at those in terms of sequence, and based on similarity, which one originated from where, which one originated from which strain. So we can actually look at, so it's also the phylogenetic analysis assists in epidemiology a lot of infectious diseases like COVID, or genetic defects, how it spreads in a certain region. So it has many different uses. Also, it may aid in prediction of function of new novel genes. You may use it in biodiversity studies and understanding microbial ecology. So this is, it has many uses. Mitochondrial DNA, we have to mention that because it's used a lot in this family of the evolutionary descent type of thing. So if you mitochondrial DNA is inherited from maternal DNA. So sperms do not contain mitochondria. So the embryo gets the mitochondrial DNA from the mother. So we should, other than the mutations, our mitochondrial DNA should be exactly the same as our mother's mitochondrial DNA. So the maternal lineage could be traced by using, for example, this mitochondrial DNA. So it's being used a lot also in phylogenetic analysis. As I said, we can build trees by comparing different virus strains and finding their similarities between them, because these are biological sequences. You can try to sequence align them, find the sequence identity, or similarity score of these sequences, and based on that you can build trees like that show the relationship between those strains, again hierarchically. In all these trees, you see there are different parameters of these trees. For example, this tree does not seem to have a root. For example, this one has a root, or this one. There is a root here. So there's the root of the tree is here, and the branching starts from here. So there's a root, and there are levels of these branches. But here in this one, there is no root. You can take any one part, or for example, is this the root of the tree or root of the tree? So you can take any one of those, or any where in the branch could be taken out, pulled out, and selected as the root. So we can have rooted trees or unrooted trees. Another thing is that the branch lengths may mean something. For example, the reason that this variant is far away from the others is probably due to the fact that its sequence similarity is very small. It's really divergent compared to the other ones. This means that we can also assign edge weights or distances or some values to these edges, and we can draw them proportional to those edge weights. They can have length proportional to those edge weights, so that it will give us also visually, will tell us how these are related. So we can have unrooted, rooted trees, weighted edges, unweighted edges. So there are different types of phylogenetic trees that we can build. And here, the closest living relatives of modern humans and genetically, if you look at the mitochondrial, this was the premolecular view, where humans were thought to be outliers in this in this list of organisms. Okay, the great apes here, chimpanzees, gorillas, and orangutans, they were clustered together, and they were thought about as 15 to 30 million years ago. We evolved from the same common ancestor. So here, if you look at genetically, however, in the light of molecular information, in the light of genetics, we see that. And if you look at the genetic similarities to build this tree, again, it's a weighted tree, as you can see, but the distance is now more smaller. We had a more 15 to 30 million years ago, now became 14 million years ago. But another thing is that the orangutans are the outliers in this group. Humans are in a cluster together with chimpanzees, bonobos, and gorillas. Gorillas also, if in this small group, gorillas are like the outliers. Okay, so it shows that genetically, bonobos and chimpanzees are more related, closely related to humans than either are to gorillas. They can also be phylogenetic analysis can also be used in the cases like this, in legal cases, where here whether the Florida dentists were the source of HIV that the patients got, certain patients of this dentist, whether they have HIV due to the incorrect, not sterilization, for example, of the needles in this dentist. And when we do, for example, for that, we need to have some controls, these controls may be people with so we are looking at the HIV strains here, these different ones have. So local controls are the ones that are not customers of this or patients of this dentist, but have the HIV, and we see that they have certain, they are different from the samples that we got from the dentist patients, and a large number of dentist patients are grouped together here. Such a phylogenetic tree may be used as scientific evidence that show that these patients got HIV from the dentist. Okay, so phylogenetic trees are really, as I said, can be used in different places. We can use build phylogenetic trees for individual genes by looking at similarities between individual genes, or we can try to build trees by looking at overall similarity measures between entire species. So I'm going to skip these parts. These are different evolutionary events that can happen. For example, if you build the phylogenetic tree for a single gene, you can see certain evolutionary events could be inferred after you have that gene, phylogenetic tree for that gene. And when we're building phylogenies, in the before we had genetic data, phenotype information was used mostly, like the traits of those species, whether they have wings or not, whether they can swim or not, things like that. These are characteristics. And phenotype information can be difficult to observe for very small species like bacteria, difficult to compare diverse species within plants within bacteria. Okay, so therefore the data we built for phenotype for genes or species these days is mostly genetic information that we can measure more accurately. Yeah, we are going to be focusing on numerical distance estimates between these species or genes, and we will be given in all the algorithms we are going to see in this chapter. We are going to assume that a distance matrix is given to us. Okay, that will be our input, and our goal will be to state this as a computational problem that I will describe next. I'm skipping this one too. I've talked about different kinds of trees. Edge lengths may mean something. There may be trees with no edge weights too. So I'm skipping these parts. We can how to give a root to uh, an unrooted tree. You're not going to be dealing with unrooted trees in our examples. Okay, they will all be, we will be having an agglomerative clustering. We will group individual items one by one here hierarchically until we have a single group of all those items, and that single group will be our root node. So I'm skipping this rooted, unrooted. Ages are weighted with a million years ago, for example. They may mean something, or they may indicate similarity. So there are different ways to have these methods. Now, so yeah, this maximum parsimony method or maximum likelihood methods are used for character-based methods in which the input is something like this. When the input is a character state matrix like this, if you have a bunch of characters or phenotypes or features, you may call them, and for each species, you have a binary value indicating whether that species has that character or not. If you have a character state matrix, there are computational methods like maximum parsimony, maximum likelihood, most of them coming from statistics to build those trees. We are going to focus on distance-based methods, UPGMA and neighbor joining. We will learn these two algorithms, and I will quickly go over the other algorithm that I mentioned in the first weeks of this course. Here's the place where we define this as a computational problem for those distance matrix methods. And here, let me go to here. Now we are given as input, we are given a distance matrix. So this distance matrix is this capital D. So this is given to us as input. This distance matrix is a square matrix which shows the relationships between a number of species. So here, if it's the distance matrix, it may contain all zeros in the diagonal. It may, I think it should be symmetric. In most of the cases, it will be symmetric. In other words, which would I mean that the distance between A and B is going to be equal to distance between B and A. Okay, so all the distance measures, similar sequence similarity measures we have seen up to now, they have this property. Whether you're comparing A and B or B and A, it doesn't matter. You're comparing two things. There is no direction in comparison or in computing the similarity or distance between two species. So therefore, it's a symmetric. The distance is symmetric, which means that all we need to be given is this, either the upper triangle or the lower triangle. These two parts will be equal to each other in terms of their mirror images will be equal to each other. Okay, so we are given a matrix like this, and our goal will be to create a tree in which these individual species A, B, C, D, E, they may be ordered differently. Maybe E is going to be here, which who knows. We are going to build a tree with by combining two of them each time. We don't know which ones to combine first. Okay, and we are going to assign edge weights to these three edges. There are going to be weights here, and our goal will be to have the tree distance. We'll have a tree distance. The tree distance between two species on this tree is equal to the summation of edge weights along the unique and simple path between two species. For example, what is the path between A and C? The path between A and C is this unique path. I call this simple path. A simple path is a path in which you don't go over the same edge over and over and again. For example, somebody could have said us that the distance between A and C is this. I go here and come back. So this is also a path from A to C, right? A path from A to C, but this is not a simple path because the same edge is used twice. So it's not a simple path. So in a tree, there's a unique simple path between any two nodes in the tree, between the root node to the leaf nodes, or between any two nodes. Now, it's actually what we do is this node is called something special for example, in competitive programming. There is this problem called LCA. LCA is an acronym for lowest common ancestor. Lowest common ancestor. Okay, so it's an interesting computational problem too if you're interested in this. It shows given two nodes in a tree, which may not be at the same level like this. What is their lowest common ancestor? So this is A is direct. This node is a parent of A. This node is parent of B, parent of C. So parent and child relationship are one distance direct relationships with a direct edge. Ancestor, this node and this node and this node. These three nodes are ancestors of A. So the ancestor of A are the nodes that come before node A to the path to the root node. So root node is the ancestor of all the nodes in the tree. All right. So this is the ancestor of C. Ancestor of C. Let me mark them with a different color, with this green color. So ancestor of C, ancestor of C, and ancestor of C. So if you look at this, the lowest common ancestor problem is finding this node as the common ancestor of A and C. And the lowest common ancestor problem in the context of programming competitions, for example, is how can you find this as fast as possible? For example, there are algorithms to find the lowest common ancestor in sublinear time, in log n time, but this is off topic. I'm just going to I'm using the terminology here for describing the tree path. What we do is the path to its lowest common ancestor is unique, and in both cases. So if you find the path to the lowest common ancestors to both of these nodes, this is the unique path, unique simple path. You can combine these two parts to get yourself a unique simple path from A to C. And the summation of the edge weights on this unique path is we call this lowercase distance between A and C. Okay, now the computational problem that I'm describing right now, let's get back to this one, is this. Minimize, so for all xy, the distance between xy, the difference between this one and this one, and its absolute value. Look at what I had written here. For all xy, so for all possible pairs of species in your tree, find the tree distance, this lowercase xy, find the distance on the tree, compare it to the distance that was given to you as input in the distance matrix, the capital D. Look at the difference between them. Take its absolute value. It means the absolute difference between these two objects, and sum this over all possible pairs. And your goal will be the computational problem, which is an optimization problem we have here, is to minimize this distance between this and that. So For example, if this distance matrix was an additive distance matrix, additive means that uh, it's like it's a metric, and it's additive means that the distance measure is a metric distance measure, which means that it's symmetric. All the values are greater than or equal to zero, other than the distance to itself. And the triangular inequality holds. Triangular inequality means that the distance, it's like this. The triangular inequality, let me write it here. If you have A, B, and C, okay, so the distance from A to C is less than or equal to, so the direct distance from A to C is less than or equal to the distance from A to B plus A to C. This is called the triangular inequality. So if you're going to some place through some node, okay, like B, if you go from A to C via node B, the total distance should be greater than or equal to, so this one should be greater than or equal to directly going from A to C. And this holds, this triangular inequality holds. I mean, the case of equality occurs only if B is on the same line as AC. If they're collinear, this equality holds. But this is for the real distances. If this distance here was the Euclidean distance between these 2 points, or if they were on a map, if these were points on a map and we measure the distance as this direct distance, B-line distance between them, the triangular inequality will always hold. But the bad thing is that triangular inequality does not hold in general for sequence similarity scores. For example, if you're doing local sequence similarity, the fact that A is closer to B and B is closer to C does not mean that A and C should also be close overall, or it may be even larger. So what I'm saying is that the distance that we may be given, that matrix that we are given here as input, sometimes may not satisfy that triangular inequality. Even in that case, our goal will be to minimize this. The problem is a simple problem. When the triangular inequality holds, if we have this, if the distance is symmetric, if it's larger than or equal to zero, no negative scores, if it's zero for if the distance of the object to itself is zero, and it's greater than zero if they are different objects, and if it's symmetric and the triangular inequality holds, this problem becomes not so interesting problem, because there is always a tree in which this total difference is zero. It's always possible to come up with a tree, because tree distances, the distances on the trees, when we have positive edgeways like this, tree distances are like distances on a map. Therefore, tree distances, this lowercase d itself, has always triangular inequality. It's a matrix. The problem is when the given input is not like that, it may be impossible so to create such a tree. So what I'm saying is that if the uppercase D is already a metric, it satisfies these properties, there will always be a tree with this distance equal to zero. But if not, it will be impossible to have such a tree, because if it's not a metric, and since these three distances are metric distances, then you will never it will be impossible to get these lowercase d values that are exactly equal to these uppercase d distance values. In that case, the problem becomes more interesting because although we cannot exactly match the given input matrix, our goal will be an optimization function. Match D as close as possible. Minimizing this difference means that create a tree with these metric distances on it, such a way that it most resembles, it's as close as possible it can get to this uppercase D. So this is a difficult problem. It's actually shown to be an NP-complete problem, or maybe NP-hard, I'm not sure. So I don't know the decision version of this problem, but it's a difficult problem. So which means that there is none of these methods that we are going to see is going to guarantee that to minimize this. Okay, minimization will not be guaranteed, but they're good heuristics. Both UPGMA and neighbor joining algorithm we are going to see, they are good heuristics which which get close to the value of this input matrix D, but they will never guarantee to find the optimum tree. All right, even neighbor joining doesn't guarantee that. But the good thing is that they run really efficiently. They are really fast algorithms, and they will most of the time will give good results that will approach this original given input matrix. Okay, so this you understand this computational problem, right? So our actually what we are minimizing here, instead of using the absolute value on the slide, as you can see, the square is taken because trying to minimize a squared function like this, absolute value may be more difficult to analyze mathematically. But if you take the square of the difference between these two things, you're always going to get positive results. So usually this is called least squares error. So the deviation between our edge weight tree paths in the constructed tree and the one in the input matrix, the difference between them, you take the square, you sum them up for over all xy, and that's our least square error or least square deviation from the input. And yeah, it's an NP-complete problem, proven to be an NP-complete problem, which means that nobody has found yet a tractable solution, a solution that is practically possible to finish in some time if the number of species that we are trying to work on is large enough. Okay, but there are heuristics. They are usually and they're based on agglomerative clustering, which means that we start with individual groups and group by group we connect them together into larger groups. And UPGMA and neighbor joining are like that. And they both assume the additive distance matrices. Now this is an example tree. This is an example input matrix given to us, which is additive. In order to prove that the triangular inequality holds, you need to look at all the triplets of objects here. For example, ABC, ABD, ABE, BCD, BCE, all triplets. You have to look at them and see the distances, add them up, see whether it's really larger than the individual distance here. Okay, so the triangular inequality holds in this case. If this is the given input matrix, look at this tree on the left-hand side. This is a tree in which all three, and this is the unique tree, by the way. This is the unique tree, which is, it may not be unique if we have all the same values here. For this example, it's the unique tree. If you compute the tree path on this tree, you'll see that it gives you the distance value that is given to you in the matrix. For example, from E to B. What is the distance from E to B? The distance from E to B is 14. Let's see whether it's 14 here. 5.5 plus 2.5, it's 8, 11, and 13, and 14. So we get distance 14. So pick any two leaf nodes, compute the distance, tree path distance between them. For example, between B and C, it's 4. B and C, it's 4. Between B and D, it's also 4. They are not in the same branch, but if you look at the distance between B and D, it's also 4. 1, 3, and 4, it's also 4. Okay, so there always exists a tree when we are given an additive distance matrix. An additive distance, as I repeat, is a matrix which is symmetric, satisfies triangular inequality. The distance is 0 only if two objects are equal to each other. The distance to itself is 0, and otherwise the distance is greater than 0. So if we have a distance matrix that satisfies these properties, we call it an additive matrix, and there is always a tree possible exactly matching the values here. So in the case of additive distance matrices, we don't have an NP complete problem. We have an algorithm that I described in the first week of the course that actually creates this tree. We can do that right now for this one. Let me quickly copy and paste it. And in the last 10 minutes, I'm going to go over that method quickly again. And we are going to talk about UPGM and neighbor joining on our Monday lecture after the spring break. So we won't be meeting this Monday because we have spring break and there's Ramazan Bayramı. So I'm going to be meeting you in about 10 days or so. Okay, so let me scale this down a little bit. Okay, this is actually, I only need the matrix, but let's leave this here also as a reference. Scale. Let's move it here. And let me scale, flatten my image. Okay. Now, start with, the algorithm is like this. Start with any pair of nodes. For example, let's start with CNA. Okay. And what we're going to be doing is that's CNA, they form the first branch in our tree, and whatever the distance we have between them. So C here, A here, what the distance between C and A is 12. So I write 12 here. So this is the first step. Then I pick a third node, again randomly, to branch off from this existing branch between C and A. Okay, so let's pick, for example, D. Let's pick D. So what I'm going to be doing here is that I'm going to make D branch of some point here. The thing that is not known at this point is, let me also erase this 12. I'm going to write it here at the top. Okay, now the whole distance is 12, but I don't know what this X, Y, and Z going to be. Okay, so let me write, you can label them anyway. Okay, so there are three unknowns here, three unknowns, X, Y, Z. But the thing is that we have three equations that come from the distance matrix to us. We already know that X plus Y is equal to 12. X plus Z is the distance between, X plus Z is the distance between these two, C and D. What is the distance between C and D? 6. And Y plus Z is equal to the distance between A and D, which is 8. So three unknowns, three equations, we solve this. If we do that, for example, X minus Y is equal to -2. Combining these two equations, subtracting the second one from the first, I can get rid of Z. Okay, so in other words, I can have X minus Y is equal to -2, and X plus Y was equal to 12. I can add these two equations to get myself 2x is equal to 10 and x is equal to 5. So if x is equal to 5, you just write 5 here. Then this part becomes 7, and z becomes what? X plus z was 6, z becomes 1, right? And y plus z was 8. Yeah, it's 8. As you can see, everything is satisfied right now. Between A, C, and D, we have this tree. Now we continue doing this until all the objects are added one by one. Let me change the color now to the red color. We can try to add anything between any two objects here. So we are going to select an object that is not added yet, B or E. Okay, we are going to again select one of them randomly, and we are going to try to insert them to any random pair we choose from here. For example, we can choose to insert them between A and D, C and D, or A and C. Okay, so let's try to do that again between A and C if you want. But what we need to be careful in this algorithm that after we find that the branching, for example, let's do that again here this time. Let's try to branch for example, b from here. Okay, let's try to do that here. And if the value of x, this time x is going to be here, the x that I define is going to be here, and y is going to be here, and this is going to be z. Okay, if this x also turns out to be 5, which means that if that x turns out to be 5, this b is going to be also branching from the same node. This is a problem for us because it's going to mess up the triangular inequality thing. We don't want to be branch off from this an existing branch. If that happens, instead of trying to insert b between c and a, we try this again, try to insert b again between a different pair, maybe between c and d the next time. So let's see what will happen when we try to add b between a and c. Okay, so x is here, so this is x, this is y, this is z. So x plus y is again 12. X plus z is the distance between b and c, which is 4. And y plus z is the distance between a and b, which is 10. Okay, so let's try to do it again. Again, I'm going to subtract this one from this. I'm going to use these, subtract this one with -1. X minus y is equal to -6 this time. 10 minus 4 minus 10 is -6. And x plus y is equal to 12, which gives me 2x is equal to 6, x is equal to 3. Okay, so what does this tell me? This tells me that the only thing, there's no problem, okay, but it also tells me that the figure that you drawn here is not actually visually not correct. Okay, if x is 3, b actually should be branching somewhere here because the distance from here to here was 5. If x is 3 in your equation and y is equal to 9 in this case, so this is a larger distance. So only you have drawn b in the wrong place, but this is not important. Okay, we don't need to undo anything. All we need to do is erase this part and say, okay, b has to branch here. This time you will update certain distances. This 5, this part is going to be 3. The branch between this intermediate node and this intermediate node is going to be 2, because 3 plus 2, from here to here it was 5. So this place becomes 2. And there, since this distance is 9 in total, so again 2 is, so it was 7, right? So here 2 is going to be here, and 7 is this location. So sorry, actually, so 3, 5, yes, 7 is going to be this entire location here. The entire thing is going to be 7. This branch is not there, so it's confusing me, so I need to erase that part. So this branch you see here is not there. We put b over here to be visually correct. Okay, so so you find your y actually becomes this one, which is also true, right? 2 plus 7, it's also 9. It's true. The only thing is on this branch, this branching from b is 3 distance away from c. The distance between these two branches is 2, and the distance from this branch to a is 7. So the total is still 12. The distance from this branch to a is still 9. This was our y in this case. Okay, so it's 9. This one is 9. And the distance from, so y plus z was 10. Z is therefore 1, right? Oh, that's a problem then. Let's see. If z is equal to 1, is it a problem? This was, yeah, for our case, z is like 3, right? So so if z is equal, if the distance between, so let's see, this was 3. So if this is x, this is y, and this is our z, that's So, or sorry, yeah, actually it is. If it is 9, then z has to be equal to 1. Then this is not, this is a problem. We cannot have, in our case, rz here is the distance from this branch. Oh, no, 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 we are not talking about d at all. Rz here is this one. So this branch here, from here to here, we'll have 1. This is exactly saying that, no problem. Somehow I was confused. I put this d into picture. In this system of equations, the species d was this branch we don't consider at all. Okay, we were trying to add b between a and c. Therefore, the z that we defined, which I erased here, was the distance of b from this branch here. So I'm going to put 1 here, which makes everything. So x plus z is equal to 4 is okay. 3 plus 1. This x plus y is equal to 12 is again satisfied. 3 plus 9. And also y plus z is equal to 10 is again satisfied. This place here is y. This place here is z. So 9 plus 1 is equal to 10 is again satisfied. Okay, so the adding of this was not at a branch, so we were able to add this nicely. We do the same thing for E, inserted between any chosen between A and B or A and D or A, C and D, whatever pairs of objects you choose. You try to add E the same way by solving this 3 equations with 3 unknowns. And at the end, you're going to see you will end up with this exact same 3 if you try to solve it for E also. Actually, I can quickly do that. For example, let's try to add E between A and D. Okay, so here if you try to add between A and D, here are the things that we are going to define. Let's call this one. Let me use a different color again. Green one. I will quickly do that and conclude this lecture. So if we try to insert E here, this becomes, let's call this Y, let's call this X, and let's call this Z. This time X plus Z is equal to 7, right? The distance between A and E. The distance between A and D is 8. So X plus Y is equal to 8. And Y plus Z, the distance between D and E, is 12. Again, if you subtract these from each other, X minus Y is going to be minus 4, and 2X is going to be 3. Okay, so again, I multiplied this with minus 1. X minus 1 became minus 4. X minus 1 is equal to minus 4. X minus Y is equal to minus 4. I added the, oh, sorry. I cannot, so I gave them different things. So I need to get rid of Y in this case. So yeah, if you multiply this, yeah, exactly, you're getting rid of Y if you add these two, not X minus Y, it's going to be X minus Z. If you multiply this with minus 1, Y's cancel out each other, this one becomes X minus Z. X minus Z is equal to minus 4. If you add these two equations, 2x becomes 3, then x is equal to 1.5. If x is equal to 1.5, which is here, this part here becomes the distance between A and E, what was it? 7. 7 minus 1.5 becomes 5.5. Okay, and this entire y distance here, so if this is 1.5, you do the math. But as you can see, we got the exact same 3 where the distance from A to this intermediate node was 1.5 and to A it was 5.5. So the exact same 3 we were able to obtain it by using this algorithm, which is iterative, but this algorithm only works correctly if we have an additive distance matrix. It's a simple, fast algorithm, but given an additive distance matrix, it works exactly and correctly. All right, now I will stop here and we are going to see 2 methods. So we have finished this one. UPGMA and neighbor joining will work on any matrix. They will work on additive or non-additive. They will work on any matrix that is given to us and build a tree out of that. but it means that they will not be resulting in a tree like this. For example, if you apply the UPGML algorithm on this matrix, you will get a really different tree than this one. So it will not be the optimal tree. All right, any questions? All right, if not, I will stop recording now.